Tetrahydrocannabinol, or THC, is an active exogenous compound that alters human perception. Regular consumption results in a specific physiological consequence, an artificially elevated sensation of pleasure. This sensation is driven by a surge in dopamine at certain synapses within the cerebrum. At high dosages, the manipulation of these neural pathways becomes extreme enough to trigger hallucinations. These macro-level behaviors are only understood by mapping the exact microscopic pathways they hijack. Common pharmacological assumptions suggest that a drug causing pleasure must directly stimulate the brain's reward centers. Empirical data reveals a biological contradiction. THC does not stimulate dopamine neurons. Its primary action is the suppression of chemical signaling. This raises an investigative question. How can suppressing a neural signal result in a massive surge of dopamine? Solving this paradox requires understanding a neurobiological principle called disinhibition. In complex biological networks, a compound often turns a system on by targeting and turning off its internal regulators. We begin with the generic synaptic cleft, the microscopic gap where all neurological communication occurs. Communication follows a 5-step sequence. Step 1 is the arrival of the electrical nervous message at the terminal buds of the presynaptic neuron. Step 2 is exocytosis, where vesicles fuse with the membrane to liberate neurotransmitters into the synaptic cleft. Step 3 is the fixation of these neurotransmitters to specific receptors on the postsynaptic membrane. Step 4 is the generation of a postsynaptic membrane potential, allowing the nervous message to continue. Finally, Step 5 requires the elimination of neurotransmitter molecules through enzymatic degradation or recapture by the presynaptic membrane. Every human sensation relies on this uncorrupted electrochemical sequence functioning perfectly. To find where THC breaks this chain, we look at a specific cell type in the cerebrum, the GABA-releasing neuron. In Study 1, researchers measured the quantity of GABA released before and after electrical stimulation, both with and without THC. This data table presents the experimental results. Without THC, the baseline GABA release before stimulation is null. After stimulation, the control group releases a big quantity of GABA. Now we introduce the exogenous variable to observe how THC binds to and alters this neuron's function. According to the with THC row, even after stimulation, the presence of THC results in only a small quantity of GABA being released. The data proves THC is an inhibitor that restricts the vital exocytosis step in GABA neurons. Now pivot to the next node in the cerebral network, the dopamine-releasing neuron. This is our target molecule responsible for the sensation of pleasure. Study 2 maps how the GABA levels we just measured impact the adjacent dopamine neuron. This schematic traces the physical relationship between the two neurotransmitters. When the upstream neuron releases a big quantity of GABA, represented by the red squares, it binds to the dopamine neuron. This heavy barrage of GABA results in a small quantity of dopamine being released. The inverse scenario follows a strict mathematical pattern. On the right, the upstream neuron releases a small quantity of GABA. Lacking the red squares, the downstream neuron fires heavily, releasing a big quantity of dopamine. This visual evidence establishes that GABA and dopamine exist in a strict inverse proportion. GABA acts as an inhibitory neurotransmitter, the neurobiological brake designed to suppress dopamine release. Combining these variables reveals a single dynamic 3-node circuit. First, the exogenous THC molecule binds to the receptors on the GABA-releasing neuron. This binding chokes off the neuron's activity, restricting GABA exocytosis into the first cleft. The downstream dopamine neuron is now starved of its normal inhibitory GABA signals. Lacking its biological brakes, the unrestrained dopamine neuron fires heavily, flooding the final cleft. This sequential logic, inhibiting an inhibitor to create a positive output, is the principle of disinhibition. This fundamentally solves our paradox. THC generates pleasure by successfully disabling the internal chemical mechanism that normally limits it. Understanding pharmacology requires looking past macro symptoms to decode the microcircuitry. Disinhibition serves as a foundational architectural model for how a wide range of exogenous compounds manipulate human neurochemistry. Scientific clarity comes from mapping the exact biological step where a compound breaks the chain of standard transmission.